Welcome to the Deep Dive. Today, we're plunging into something absolutely fundamental for you, the cell. Think of it as the ultimate building block, the very essence of life. We've got a great collection of information here that really breaks down what these tiny powerhouses are all about, their structure, how they function, and their life cycles.
Yeah.
Our mission in this deep dive, tailored specifically for you, is to extract the core concepts that are going to be so crucial in your nursing studies. We're talking about laying a solid groundwork for understanding how medications work and how disease processes impact the body at the most basic cellular level.
Absolutely. Understanding the cell isn't just about, memorizing diagrams. It's about grasping the why behind so many biological events you'll encounter.
Right.
When you truly understand what's happening at the cellular level, things like pharmacology and pathology will start to make a much deeper and more practical sense for you.
Okay, so to give you a clear roadmap for our deep dive today, we're going to start by looking at the cell's main components, the nucleus, that all-important control center.
The brain, really.
Yeah, the brain. Then the cell membrane, our incredibly selective gatekeepers, and the cytoplasm, the bustling interior where, well, so much happens. Then we'll explore the fascinating world of organelles, those tiny organs within the cell, each with a very specific job. We'll also zero in on the unique properties of the cell membrane and how substances manage to cross this crucial barrier. Finally, we'll touch on the different phases of the cell's life cycle, a cycle that's essential for growth and repair in the body.
And remember, it's all interconnected. How a cell is structured directly influences its function, and understanding these basics will really serve you well as you encounter more complex topics in your coursework. You'll begin to see how drugs you administer might target very specific cellular components or processes, and how diseases you'll be managing can disrupt normal cellular activity in profound ways.
All right, let's unpack this, starting with the very basic structure. The cell, as we said, is the fundamental structural unit of the body. It's how your patient's tissues and organs are formed. And every cell, with a few exceptions, has those three main parts we mentioned. First up is the nucleus. Think of this as the cell's command center. And what makes the nucleus so critically important for you to understand?
Well, it's the repository of our genes, which are made of deoxyribonucleic acid, or DNA. DNA contains all the instructions for making proteins and directing every aspect of cell function. It's made up of nucleotides, and these have a deoxyribose sugar, a phosphate group, and one of 4 nitrogenous bases: thymine, cytosine, adenine, and guanine.
Okay.
These bases, in their specific sequences, hold the genetic code, the blueprint of the cell.
So it's like a blueprint written in a 4-letter alphabet that dictates everything the cell does. And this blueprint directly guides the production of proteins. Why are proteins so essential for cellular function? Something you'll see reflected in so many disease processes.
Oh, proteins are truly the workhorses of the cell. They carry out a vast array of functions that are critical for life, from catalyzing biochemical reactions as enzymes to providing structural support. The DNA in the nucleus holds the master instructions, but to actually make these proteins, we need another key player, ribonucleic acid, or RNA.
Okay, so how does RNA fit into this essential protein production process?
Well, RNA is similar to DNA, but it's usually single-stranded. It has a ribose sugar instead of deoxyribose, and it uses uracil instead of thymine as one of its bases.
Oh, right, different base.
Exactly. The process starts with transcription, where the information in DNA is copied into messenger RNA or mRNA. Think of mRNA as a disposable copy of a specific set of instructions.
Okay.
This mRNA then leaves the nucleus and travels to the ribosomes in the cytoplasm, where the actual protein synthesis or translation takes place. Within the nucleus itself, we also have the nucleolus, a dense area that plays a vital role in forming those ribosomes, the protein-making machinery.
Fascinating. So the nucleus is this protected vault for our genetic information, and it carefully sends out instructions via mRNA to be executed in the cytoplasm. Now, let's talk about that outer boundary, the cell membrane. What's its primary role in maintaining the cell's health and interacting with its environment? Something you'll be constantly considering in patient care.
Yeah, the cell membrane is a thin but incredibly vital barrier that separates the inside of the cell, the intracellular fluid, from the outside environment, the extracellular fluid. It's absolutely essential for maintaining the cell's physical integrity and its internal stability, or homeostasis, that delicate balance you'll be working to support in your patients.
And I remember reading that it has this really unique structure. It's not just a solid wall, is it?
No, not at all. It's described as a lipoprotein structure, primarily made up of phospholipids, glycolipids, and cholesterol.
Lipoprotein.
The phospholipids are the main component, and they have this really interesting bipolar arrangement. They have a hydrophilic or water-loving head and two hydrophobic or water-fearing tails.
So they line up with their heads facing the watery environments inside and outside the cell, and their tails meet in the middle, creating this oily barrier.
That sounds crucial for controlling what gets in and out.
Precisely. This arrangement is key to regulating what can pass in and out of the cell. Water-soluble substances will have difficulty getting through that hydrophobic interior, while lipid-soluble substances can pass more easily. This has significant implications for how drugs are absorbed and distributed in the body, something you'll learn a lot about. The membrane is also fluid, meaning its components are constantly moving, which allows the cell to change shape and even repair itself if it gets damaged.
That's incredible. It's dynamic and self-healing.
Yeah.
And you mentioned glycolipids and cholesterol. What specific roles do they play in the membrane's function?
Well, glycolipids, which are phospholipids with a carbohydrate chain attached, are found on the outer surface of the membrane and can be involved in cell recognition and signaling, essentially allowing cells to communicate with each other and identify foreign invaders.
Like little flags. Kind of, yeah. And cholesterol is interspersed within the phospholipid bilayer and helps to stabilize the membrane, keeping it fluid but also preventing it from becoming too permeable. Think of it as ensuring the membrane isn't too stiff or too floppy.
Okay, so the membrane is this active selective barrier. Now, what's going on inside the cell within this membrane? That's where the cytoplasm comes in, right?
That's right. The cytoplasm is everything within the cell membrane, excluding the nucleus. It's a gel-like substance and the site of most of the cell's metabolic activities and specialized functions. And within the cytoplasm, we find those important structures called organelles, the tiny organs of the cell, each with a dedicated role.
Organelles, the tiny organs of the cell. Let's dive into some of the key players here. First up, mitochondria. I always think of them as the powerhouses of the cell. Why is that analogy so fitting?
That's a perfect analogy, and it's one you'll hear often. Mitochondria are rod-shaped, double-membraned organelles responsible for generating most of the cell's energy in the form of adenosine triphosphate, or ATP. ATP is like the cell's energy currency, fueling all its activities, from muscle contraction to nerve impulse transmission.
And I recall that cells that have high energy demands have a greater number of mitochondria. That makes intuitive sense.
Exactly. Think about cardiac muscle cells, which are constantly contracting to pump blood throughout the body. They are absolutely packed with mitochondria.
Right.
Conversely, cells that are less metabolically active have fewer. Interestingly, mitochondria can even reproduce themselves based on the cell's energy demands. For example, milk-producing cells in mammary glands will significantly increase their number of mitochondria when lactation begins, requiring a surge in energy production.
So how do these cellular powerhouses actually go about producing ATP? What's the process?
They take in carbohydrates, fats, and proteins from the cytoplasm and break them down through a series of biochemical pathways, including the citric acid cycle and oxidative phosphorylation. This last step, oxidative phosphorylation, is particularly crucial because it requires oxygen.
Oxygen. Key ingredient.
Absolutely vital. This is why oxygen delivery is so vital in patient care. Without it, ATP production suffers. When oxygen is scarce, the mitochondria can't produce as much ATP, and the cell has to rely on less efficient anaerobic respiration, which leads to the buildup of lactic acid. This lactic acid then gets transported to the liver for processing.
That explains why we see elevated lactic acid levels in patients experiencing hypoxia or shock. Okay, next up, the endoplasmic reticulum, or ER. This sounds like a vast internal network within the cell.
It is. The endoplasmic reticulum is an extensive network of interconnected channels, or sacs, called cisternae, that spread throughout the cytoplasm, forming a huge surface area for various chemical reactions to take place within the cell.
Huge surface area.
You distinguish between two main types, rough ER and smooth ER.
What are the key differences between the rough and smooth ER, and how do those differences relate to their functions?
Well, the rough ER has ribosomes attached to its surface, giving it a rough appearance under a microscope. These ribosomes are the primary sites of protein synthesis.
Ribosomes, okay.
So the rough ER is heavily involved in producing proteins that are destined for secretion outside the cell, insertion into the cell membrane itself, like those receptor sites we discussed, or delivery to other organelles within the cell. It also plays a role in producing phospholipids and cholesterol, key components of cell membranes. The smooth ER, on the other hand, lacks these ribosomes, hence its smooth appearance. It's involved in a more diverse range of functions, including further lipid and cholesterol production, hormone synthesis in some cells, like steroid hormones, and very importantly for you to remember, the detoxification of certain drugs and toxic substances you might encounter in your patients.
So the rough ER is like the main protein manufacturing plant with all the machinery, the ribosomes, and the smooth ER has more varied roles, including detoxification, something critically important when considering how the liver metabolizes medications. Now, we've mentioned ribosomes a couple of times. Let's focus on them directly. What exactly do these crucial structures do?
Ribosomes are the molecular workbenches where proteins are assembled according to the genetic instructions. They read the code carried by messenger RNA, mRNA, which, as we discussed, originated from the DNA in the nucleus.
Right. Reading the mRNA code.
Ribosomes themselves are made of ribosomal RNA, and proteins. We find them in two main locations. Some are attached to the rough ER, where they synthesize proteins for specific destinations, while others float freely in the cytoplasm, synthesizing proteins that will be used within the cell itself, such as structural proteins and some enzymes needed for metabolic processes.
And the transfer RNA, or tRNA, that we briefly mentioned earlier, that's the molecule responsible for bringing the correct building blocks to the ribosomes, right?
Exactly. Transfer RNA, tRNA molecules act like tiny delivery trucks, each carrying a specific amino acid, the building blocks of proteins.
Amino acids, got it.
They read the mRNA sequence in the ribosome and ensure that the amino acids are added in the correct order, like following a precise recipe to build the specific protein the cell needs.
This sounds like an incredibly complex and highly coordinated construction process happening constantly within every single cell in the body. Okay, another organelle I remember is the Golgi apparatus. What's its role in this intricate cellular factory?
The Golgi apparatus is often likened to the cell's post office or distribution center.
Post office, okay.
It's a series of flattened, membrane-bound sacs called cisternae. Its main job is to further process, modify, sort, and package proteins and other substances that have been synthesized in the ER. Think of it as adding the finishing touches and addressing labels to the products.
Like gift wrapping.
Sort of, yeah. These finished products are then sorted and packaged into small membrane-bound sacs called vesicles, which can then be transported to other parts of the cell or secreted outside the cell through exocytosis. The Golgi might also be involved in producing lysosomes, which we'll discuss next, and storing synthesized proteins and enzymes until they are needed.
So the ER builds and initially modifies the molecules, and the Golgi then puts the final touches on them, packages them, and sends them to their correct destinations, either within or outside the cell. That makes perfect sense in terms of cellular organization. Now let's talk about lysosomes. These sound like the cell's internal cleanup crew and defense system.
That's a very accurate way to think of them. Lysosomes are membrane-bound organelles that contain a powerful cocktail of hydrolytic enzymes, essentially digestive enzymes.
Digestive enzymes. Strong stuff.
Very. These enzymes are capable of breaking down all sorts of cellular waste, including proteins, nucleic acids, carbohydrates, and lipids. They play a crucial role in digesting worn-out cell parts, recycling their components, and even in the controlled destruction of the entire cell when its membrane ruptures, a process called apoptosis, or programmed cell death.
So they're involved in both the routine maintenance and the more drastic measure of cell death. That sounds important in both normal tissue turnover and in pathological conditions.
Exactly. Lysosomes form a membrane around whatever needs to be digested and release their enzymes into that isolated area, protecting the rest of the cytoplasm from these potent enzymes. You can see a somewhat analogous process on a macroscopic scale with decaying organic matter like lettuce left in the fridge too long.
Yeah, it just kind of digests itself.
Exactly. That breakdown is facilitated by similar enzymatic actions. This process is also critically important in the breakdown of bacteria and other foreign materials engulfed by immune cells.
That really highlights their role in the body's defense mechanisms. And you mentioned that lysosomes can have clinical significance beyond just normal cell function. How so, particularly in the context of nursing care?
Well, when cells die due to injury, disease, or even the effects of certain drugs, the released lysosomes can potentially damage neighboring healthy cells by breaking down their essential proteins and membranes.
Ooh, collateral damage.
Right. This can lead to a chain reaction of tissue damage, as we see in conditions like pressure ulcers or bed sores, where localized cell death can spread and worsen the wound. Debridement, the removal of dead and damaged tissue, is often a crucial part of managing these ulcers because it helps to remove the source of those released lysosomes and allows healthy tissue to regenerate. Understanding this at a cellular level helps explain why debridement is so important.
That makes a lot of sense. Removing the damaged tissue stops the cascade of destructive enzymes.
Okay.
Okay, we've explored the main organelles and their vital functions. Now, let's circle back to the cell membrane and some of its unique properties beyond just acting as a physical barrier. I know it has these things called receptor sites. What are they all about, and why should this be important for someone going into nursing?
Receptor sites are specific peripheral proteins embedded in or associated with the cell membrane. They act like highly specific docking stations that can bind to particular chemical messengers outside the cell, such as hormones, neurotransmitters, or even certain drugs you might administer.
Yeah.
This binding then triggers a cascade of events inside the cell, leading to a specific intracellular reaction.
So it's a very precise lock and key mechanism. A specific molecule fits a specific receptor and initiates a very specific response within the cell.
Exactly. For example, the receptor site for insulin on a cell's surface binds to insulin in the bloodstream, which then signals the cell to increase its uptake of glucose from the blood. This is fundamental to understanding diabetes and its management.
Crucial.
Receptor sites are crucial for communication between cells and for how cells respond to their environment. They are particularly important in neurons, muscle cells, and endocrine glands, and as you'll learn in pharmacology, they are also key targets for many medications. For instance, beta blockers, a class of drugs used to treat high blood pressure, work by blocking specific adrenergic receptors on heart muscle cells.
Interesting. So drugs can be designed to either activate or block these receptors, essentially manipulating cellular activity. Another property of the cell membrane you mentioned was identifying markers. What's their purpose, and how does this relate to the immune system?
These are surface proteins, also known as histocompatibility antigens or human leukocyte antigens, HLA. They are genetically determined markers that essentially act as a cellular fingerprint, identifying a cell as belonging to a specific individual, a self cell.
It's like a cellular ID card that says, I belong here.
Precisely. The immune system, particularly immune cells like T cells, constantly survey the body's cells, checking these markers.
Like tiny security guards.
Yeah. If they encounter a cell that doesn't have the correct self-markers, such as a virus-infected cell or a foreign cell from a transplanted organ, they recognize it as non-self and mount an immune response to destroy it. This is why matching these antigens is so critical in organ transplantation to minimize the risk of the recipient's body rejecting the donated organ as foreign.
And I can imagine if these markers get altered or are not recognized properly, it could lead to significant health issues.
Absolutely. Cell injury, viral infections, aging, and other factors can potentially change these antigens. If the immune system fails to recognize altered self cells, it might allow abnormal cells, like cancer cells, neoplasms, to grow unchecked. Conversely, if the immune system mistakenly identifies normal self cells as foreign, it can attack them, leading to autoimmune disorders and chronic inflammatory conditions, such as rheumatoid arthritis or lupus.
That really highlights the critical importance of these seemingly small surface proteins in maintaining health and preventing disease. The last unique property of the cell membrane we wanted to touch on is channels or pores. What do they allow, and how might this be relevant in patient care?
These are protein channels that span the cell membrane, creating water-filled pathways or tunnels that allow small water-soluble substances, primarily ions and water, to move in or out of the cell down their concentration gradients.
So specific tunnels for specific things.
Exactly. We have specific channels for various ions like sodium, potassium, calcium, chloride, and bicarbonate, as well as aquaporins, which are specialized channels specifically for the movement of water in and out of cells.
And just like with receptors, these channels can also be key targets for certain medications.
Yes, indeed. Many medications are designed to specifically block or modulate the activity of these ion channels, thereby affecting the movement of ions and altering cellular function. A good example, as we mentioned earlier, is calcium channel blockers like amlodipine,
Right.
which are used to treat high blood pressure and other cardiovascular conditions by preventing calcium ions from entering certain muscle cells in the heart and blood vessels, leading to relaxation and vasodilation. Understanding which channels are affected by different drugs is a core part of pharmacology.
Okay, so the cell membrane is far more than just a passive barrier. It's actively involved in communication, identification, and the controlled movement of substances. Speaking of movement, let's delve into the specific ways substances actually move across this membrane. We have endocytosis and exocytosis, which seem like fundamentally opposite processes.
They are indeed opposite, but equally important for cellular function. Endocytosis is the process by which the cell engulfs substances from its external environment and brings them inside by extending its cell membrane around them, eventually forming a vesicle inside the cell.
Engulfing. Okay.
There are two main types you should know about: pinocytosis and phagocytosis.
Pinocytosis, I often hear that described as cell drinking. Is that an accurate, easy-to-remember analogy?
That's a common and quite helpful analogy, yeah. Pinocytosis, or cell drinking, involves the cell taking in small droplets of extracellular fluid containing dissolved substances, often including specific molecules that have bound to receptor sites on the membrane, triggering their uptake.
So it can be triggered.
It can be. It's a non-selective process for fluids, but can be selective for molecules that bind to specific receptors. It's a way for cells to sample their environment and absorb nutrients, enzymes, and other needed materials. Phagocytosis, on the other hand, is often referred to as cell eating.
Cell eating. Big stuff.
Right. It's a more selective process used by specialized immune cells like neutrophils and macrophages to engulf and destroy larger particles such as bacteria, foreign particles, and cellular debris. They essentially extend their cytoplasm to surround the target and then enclose it in a large vesicle called a phagosome, where digestive enzymes from lysosomes can then break it down.
So pinocytosis is more for taking in fluids and dissolved solutes, while phagocytosis is for engulfing and eliminating larger particulate matter. And exocytosis is essentially the reverse of this.
Exactly. Exocytosis is the process by which cells expel substances from their interior to the outside. Vesicles containing hormones, neurotransmitters, enzymes, waste products, and other materials that need to be secreted or eliminated move toward the cell membrane, fuse with it, and release their contents into the extracellular space.
Getting rid of stuff.
Or sending out signals. Think of how nerve cells release neurotransmitters to signal to other nerve cells, or how endocrine glands release hormones into the bloodstream. These are examples of exocytosis.
Okay, so that covers how cells import and export larger molecules or particles. Now let's shift our focus to the movement of smaller substances across the membrane, which brings us to the concepts of passive and active transport. Passive transport doesn't require the cell to expend any energy, right?
Correct. Passive transport relies on the natural kinetic energy of molecules and their tendency to move down their concentration gradients from an area where they are more concentrated to an area where they are less concentrated. It's exactly downhill. There are three main types of passive transport that are particularly important for you to understand: diffusion, osmosis, and facilitated diffusion.
Diffusion, that's simply the movement of a solute from an area of high concentration to an area of low concentration until an equilibrium is reached.
Precisely. The steeper the concentration gradient, the faster the rate of diffusion. As we discussed earlier with the cell membrane structure, the membrane itself acts as a selective barrier to diffusion. Small, nonpolar, lipid-soluble substances can generally pass directly through the phospholipid bilayer.
Like gases.
Like gases, yeah. Carbon dioxide and oxygen exchanged in the lungs, for example. Water can also diffuse across the membrane, albeit slowly in some cases, unless there are aquaporins. Interestingly, negatively charged substances tend to move across more easily than positively charged ones due to the electrical properties of the membrane. A common example is the movement of oxygen from the high concentration in the capillaries into a metabolically active tissue cell, where the oxygen concentration is lower due to its consumption in ATP production.
Makes perfect sense, moving from where there's more to where there's less, driven by the concentration difference. Now, osmosis is also a form of passive transport, but it specifically involves the movement of water. Correct?
Yes. Osmosis is the movement of water across a selectively permeable membrane, like the cell membrane, from an area of low solute concentration, meaning high water concentration, to an area of high solute concentration, meaning low water concentration.
Water follows the solutes.
Exactly. The water is essentially trying to dilute the more concentrated solution and equalize the solute concentrations on both sides of the membrane. This movement of water generates what we call osmotic pressure.
And this osmotic pressure is what underlies the concepts of isotonic, hypertonic, and hypotonic solutions that we encounter so often in clinical practice when administering intravenous fluids.
Exactly. An isotonic solution has the same effective solute concentration as the intracellular fluid, so there is no net movement of water into or out of the cell, and the cell maintains its normal shape.
Like normal saline.
Right. A hypertonic solution has a higher effective solute concentration than the intracellular fluid, so water will move out of the cell, causing it to shrink or crenate.
Shrivel up.
Yeah. Conversely, a hypotonic solution has a lower effective solute concentration than the intracellular fluid, so water will move into the cell, causing it to swell and potentially burst, a process called lysis. Thinking about what happens to red blood cells when placed in these different types of solutions is a classic example you'll likely encounter in your studies.
That's a great visual to remember the effects of different intravenous fluids on our patient's cells. Now, the last type of passive transport is facilitated diffusion. This also doesn't require energy, but needs a bit of assistance to occur.
That's right. Facilitated diffusion is for substances that cannot easily cross the lipid bilayer on their own, often because they are polar or charged molecules, such as glucose, amino acids, and ions.
Things that don't like the oily middle.
Exactly. They require the help of a specific carrier protein or a protein channel embedded in the cell membrane to move across. The key distinction from active transport is that facilitated diffusion still follows the concentration gradient. The substance moves from high to low concentration, and it does not require the cell to expend any metabolic energy.
Still passive.
Still passive. However, this process is limited by the number of available carrier proteins or channels. Once all the carriers are occupied, the rate of transport will plateau.
So it's like having a specialized shuttle service across the membrane for specific passengers who can't cross on their own, but the shuttle only moves downhill from high to low concentration. Okay, that clarifies passive transport. Now, active transport, this is where the cell has to actively invest energy, usually in the form of ATP, to move substances across the membrane.
Precisely. Active transport allows the cell to move substances against their concentration gradient. That is, from an area of low concentration to an area of high concentration. This uphill movement requires the cell to expend energy, typically in the form of ATP, to power the transport proteins, often called pumps, involved in the process.
And what's a particularly important example of active transport that has significant clinical relevance in the human body?
The sodium-potassium pump is a classic and critically important example you'll learn a lot about.
The NAK pump.
That's the one. This pump uses the energy from ATP hydrolysis to actively transport sodium ions, No+, out of the cell and potassium ions, K+, into the cell, both against their respective concentration gradients. This continuous pumping action is crucial for maintaining the electrochemical gradient across the cell membrane, which is essential for nerve impulse transmission, muscle contraction, fluid balance, and many other fundamental cellular functions.
Wow, it does a lot.
It really does. If a cell is deprived of oxygen, ADP production significantly drops, and these active transport systems start to fail, jeopardizing the cell's ability to function and maintain its internal environment.
That really highlights how fundamentally dependent these active transport processes are on a constant supply of energy, which underscores the importance of oxygenation and circulation in our patients. And I recall reading that some medications even utilize active transport mechanisms to gain entry into specific cells.
Yes, that's absolutely correct. Some drugs are structurally similar to naturally occurring molecules that are actively transported into cells. By sort of hitching a ride on these transport proteins, the drug can reach a higher concentration inside the target cell, enhancing its therapeutic effect. Also, active transport plays a vital role in the kidneys for excreting certain drugs and for actively reabsorbing essential electrolytes and maintaining the body's acid-base balance. So understanding active transport is key not only to basic cell physiology, but also to understanding how many drugs work and how the body manages both endogenous substances and administered medications.
All right, we've covered the cell structure, the roles of its various organelles, the unique properties of the cell membrane, and the different mechanisms of transport across it. Now let's move on to the cell cycle, essentially, the life cycle of a cell. Many cells in the body are capable of reproducing themselves through a process called mitosis, right?
Yes, mitosis is a fundamental process of cell division that results in two genetically identical daughter cells.
Yeah.
This is essential for growth, tissue repair, and replacement of old or damaged cells.
Growth and repair, okay.
The rate at which different types of cells reproduce varies greatly depending on the body's needs and the specific function of the cell. For instance, cells lining the gastrointestinal tract, which are constantly exposed to abrasion and digestive enzymes, divide very rapidly.
Makes sense, high turnover.
While others, like mature nerve cells, generally do not divide at all. Various factors, such as hormones and other chemical signals, can also influence the rate of cell reproduction in different tissues. For example, erythropoietin stimulates the production of red blood cells, and various chemical signals released during infection can stimulate the production of white blood cells.
And this cycle of cell life isn't just a continuous process of division, There are distinct and carefully regulated phases that the cell goes through, correct?
Exactly. The cell cycle is a highly orchestrated sequence of events that a cell undergoes from one cell division to the next. It consists of 4 active phases: G1, Gap1, S synthesis, G2, Gap2, and M mitosis, and often a resting phase referred to as G0.
Let's break down these different phases to understand what's happening at each stage. What characterizes the G0 phase?
The G0 phase is considered a stable, non-dividing phase. Cells in G0 have exited the active cell cycle and are carrying out their specific functions but are not actively preparing to divide.
Just do their job.
Pretty much. This phase can be temporary, lasting for hours or days, or it can be permanent for some highly specialized cell types like those mature nerve cells we mentioned. As you'll learn in the context of cancer treatment, cells in the G0 phase can pose a significant challenge because many chemotherapeutic drugs are most effective at targeting actively dividing cells.
Oh, okay.
Cells in G0 are often resistant to these drugs and can later re-enter the cell cycle, potentially leading to cancer recurrence.
That explains why some cancers can be so persistent and why treatment often involves multiple rounds of chemotherapy.
Yeah.
What happens when a cell receives the signal to re-enter the cycle from the G0 phase?
It enters the G1 phase, or Gap1 phase. This phase begins when the cell receives a signal, often from growth factors or other signaling molecules, indicating that it needs to divide. G1 is a period of significant cellular activity, lasting until DNA synthesis begins in the next phase.
Getting ready.
Exactly. During G1, the cell actively synthesizes proteins, lipids, and other molecules needed for DNA replication and overall cell growth. It's essentially gathering all the necessary building blocks and energy reserves required for the more demanding phases to come.
Okay, so G1 is a period of preparation and growth. What then happens in the S phase?
The S phase stands for synthesis. This is the crucial phase during which the cell actively replicates its DNA.
Copying the blueprint.
Precisely. It's a very energy-intensive process, and the cell must meticulously copy its entire genome to ensure that each of the 2 daughter cells will receive a complete and identical set of genetic information. The cell remains in the S phase until its DNA content has doubled.
DNA replication, a truly critical step to ensure genetic continuity. What follows this duplication of the genetic material?
After DNA replication is complete, the cell enters the G2 phase, or Gap2 phase. During G2, the cell continues to grow and synthesizes the proteins required for the formation of the mitotic spindle.
A spindle, yeah.
The intricate microtubule structure that will be responsible for separating the duplicated chromosomes during the next phase, mitosis. It's the final checkpoint and preparation stage before the actual cell division occurs.
And finally, we reach the M phase. This is the phase where the actual cell division takes place.
Yes, the M phase stands for mitosis. This is the dramatic phase where the cell physically divides into 2 genetically identical daughter cells.
Splitting in 2.
Right. It's a complex process involving several distinct stages: prophase, metaphase, anaphase, and telophase, culminating in cytokinesis, the physical separation of the cytoplasm to form 2 independent cells. After the M phase, these newly formed daughter cells can then enter the G1 phase and repeat the cycle if they are destined to divide again, or they can enter the G0 resting phase to carry out their specialized functions.
So, the cell cycle is a carefully orchestrated sequence of events that ensures accurate cell reproduction, which is fundamental for tissue growth, repair, and overall homeostasis in the body. Understanding this cycle is particularly important when we consider conditions like cancer, where this tightly regulated process goes awry, leading to uncontrolled cell proliferation.
Absolutely. Many cancer therapies, including various forms of chemotherapy and radiation therapy, aim to disrupt specific phases of the cell cycle in rapidly dividing cancer cells, ultimately leading to their death or preventing their further multiplication.
Right, targeting the fast dividers.
Exactly. Understanding which phase of the cell cycle a particular cancer cell is most vulnerable to can help guide the development of more targeted and effective treatments.
Well, this has been a truly deep dive into the fundamental unit of life, the cell. Just to quickly summarize some of the key takeaways that will be most relevant for you. The cell is a remarkably intricate and dynamic unit with specialized internal structures organelles, each performing vital roles, from energy production in the mitochondria to protein synthesis by ribosomes and waste removal by lysosomes. The cell membrane is not just a passive barrier, but an active and selective interface, crucial for communication, identification, and the controlled transport of substances. Understanding the various mechanisms by which substances move across this membrane, both passively without energy expenditure, and actively. Requiring cellular energy is absolutely fundamental to understanding how nutrients get in, waste gets out, and how drugs interact with cells.
Absolutely key.
And finally, the cell cycle, with its distinct and regulated phases, underpins tissue growth and repair, and disruptions in this cycle are central to the development of diseases like cancer.
And as a future nurse, this foundational knowledge is going to be incredibly valuable in so many aspects of your practice. It will directly inform your understanding of how medications exert their therapeutic effects at a cellular level, how various physiological processes maintain health, and how pathological conditions disrupt normal cellular function, leading to disease. You'll be much better equipped to grasp the underlying mechanisms of the conditions you'll be managing, to interpret the rationale behind the treatments you'll be administering, and ultimately to provide more informed and effective patient care.
So as you continue your nursing studies, really think about how these fundamental principles of cell physiology, particularly the mechanisms of membrane transport and the regulation of the cell cycle, are implicated in the development of new diagnostic tools and therapeutic interventions for a vast range of diseases, from infections to autoimmune disorders to cancer. What specific questions do you now have about particular cell types or disease processes at the cellular level that you might want to explore further? That's your mission to carry forward. Thanks for taking the deep dive with us.